So what I'm going to talk about today is a link project which involved NIAB and most of the UK wheat breeders and some companies in Denmark and Sweden, looking at the sources of yellow rust resistance in UK and northern winter wheat varieties to get an understanding of the diversity that exists in the germplasms that breeders are working with. So this was, well, I mean, you're all familiar with yellow rust. It's a disease of maritime temperate regions. You can see from the map here it's very prevalent in the UK, France, parts of Germany, Denmark, and up into Sweden. It occurs every year, and if we didn't use fungicides, it would be a major problem. However, even in using fungicides, if you don't get the spray on at the right time, and this is a picture here courtesy of some collaborators of mine, BASF, where if you're late in applying that spray, it can have serious consequences for the amount of yellow rust you see in your crop. So the management program basically consists of growing resistant varieties as well as applying fungicides to control the disease where resistance isn't complete or starts to break down. So, as I said, this yellowhammer project was put together to characterise the sources of resistance in winter wheats from across the UK and northern Europe. And the panel was put together, assembled, which basically represents the genetic diversity over the past 60 years in winter wheats in the UK and northern Europe. It was screened by our AGT using the breeder's 35K SNP array. So we have a genotype profile of those varieties. The panel was assessed for yellow rust resistance in the field at multiple sites in the UK, France, Germany, Denmark and Sweden. And field trials were undertaken every year from 2018 to 22. So we have a massive data set. Some of the breeders scored the panel using the 1 to 9 scale, others scored using percentage infection. But what we did, we took those scales, we converted the 1 to 9 to a percentage infection and vice versa. We also transformed the percentage data using a log 10 to normalize the data. So in the end, we had something like 88 field scores. Some of the breeders scored the panel once in a season, some twice in a season. And with all the different scales, which we used for the scoring the yellow rust, we had something like 237 traits on that panel. So massive data set. This is just to represent the phenotypic diversity we saw in the panel. This is the 2022 data, and you can see that there was quite substantial variation, and it depended primarily on the location and the season. So some of these, you've got 2 scores where the breeders have scored it twice, and so the second score is later in the season when we saw more infection on the susceptible varieties in the panel. So this is just the average scores across the whole panel, just to give you an idea of the variation for yellow rust resistance that existed in this panel. If we look at the average scores between sites and between years, we can see that there was fairly good correlation between the lines in different locations and different years. But we did see that in Sweden, the correlation was less than it was to the rest of Europe. And this would possibly indicate that the pathogen population in Sweden has a different set of virulence profiles to you find in the UK, France and Germany. Okay? As you see, these are the Swedish lines, and the correlation to the UK, France and Denmark and Germany is less than if you look within those countries. But even within the UK, we found regions where the correlation wasn't 100%. Now that could be due to differences in environments in the different sites in the UK, but it also could be due to differences in the virulence, as we've seen today. You know, maybe a virulence appears in a different part of the country, a different part of the UK than to another, as we saw with the leaf rust, where we got virulence to LR24 in the West Country last year, which then spread to Yorkshire, in the West Country in 22, and then it spread to Yorkshire in 23. So some of that not perfect correlation between field trials, phenotypic data sets, is probably down to the pathogen population and differences in virulence. So for those who are not familiar with a GWAS analysis, so basically we screened, as I said, we screened all the lines within the panel using the 35K breeder SNP. We ended up with nearly 20,000 SNPs, which we then used to look for associations between a marker which defines a region on the wheat genome and whether one allele at that marker is associated with yellow rust resistance, where the other allele is associated with susceptibility. Okay? And by looking for those associations between the phenotype and the markers, you can locate regions of the wheat genome that are predicted to carry yellow rust resistance. Okay? We can locate the markers off the 35K breeders array using the Chinese spring reference genome. Okay? And then we carry out the GWAS analysis to look for the associations, and this is just an example. So if that's the wheat genome, where you get, and each dot is the marker, where you get a high association with the phenotype indicates that there's a potential resistance gene close to that marker. And we call that association a marker trait association between a marker and the phenotype. So, it's a statistical analysis. Originally, we did the analysis using a low false discovery rate. And this was so that we didn't miss any small effect resistances. However, when we finally got all that data in over those 5 years, we repeated the analysis using a more stringent cutoff, which is usually the cutoff that people publish the data. And it didn't change the QTLs we found, the marker trait associations we found. They were still quite robust. Obviously, we found a lot of marker trait associations, so we had to decide how are we going to define that this is a real marker trait association or maybe just a ghost effect. So there will be a report coming out on this project, an AHDB final report, and in that report we decided that any marker trait association that had a significance of score above the threshold of 3 at any one of those years or sites, we'd say yes, we believe that's a genuine resistance. Or if the marker trait association had a score greater than 1.5 at more than one location or more than 1 year, we called that as a genuine yellow rust resistance. In doing that, that gave us 42 marker trait associations. Okay? Of those 48 marker trait associations, we found that 29 of them, if you look at the frequency with which the resistant allele for those 29 marker trait associations occurred in that panel of 400 plus wheat varieties, 90% of the lines had the resistant allele at that marker. So for the majority of the potential resistance genes we found, they're actually fixed or nearly fixed within that germplasm, within that gene pool that represents the gene pool of winter wheats in UK and Northern Europe for the past 50 to 60 years. So the breeders have done a good job in accumulating those resistances within their gene pool. In addition, we did find 4 marker trait associations, and this is on chromosome 1A, and it's just a number that relates to the position of the marker relative to the Chinese spring reference genome. that were present at lower frequencies. So this was 18%, 17%. So these are potentially sources of yellow rust resistance that are underutilized within that gene pool. Another 6 marker trait associations had slightly higher levels of occurrence within that gene pool, but again, potential, valuable sources of resistance. I mean, I didn't want to go through the whole table of 42 marker trait associations. I've just picked out some interesting ones. There was one marker trait association on chromosome 2A, which we believe is located on the same translocation that the YR17 gene was transferred into wheat, and that's from Aegilops ventricosa. Now that marker trait association, it was effective in 2018 and 19, but became ineffective in 2020. And when I say became ineffective, it appears to be a major seedling resistance, but we lost that resistance. However, in saying that, if you look at the data from 2021 and 22, you still see a residual effect. So even though as a seedling resistance, it's no longer effective, as in the adult plants, there's a potential that a lot of these resistances still have a residual level of resistance. So by accumulating these resistances within the gene pool, which appears to be the case, you actually, it's of value to breed it. In terms of, obviously, a massive data set, we've produced an Excel file for, hopefully, breeder-friendly, where we listed all the wheat varieties and lines that were part of the panel. Along here are all the marker trait associations that were detected. The peak marker that defined that marker trait association, its chromosome, its position on the chromosome, it's the name that we've assigned to it. And then we said, is the allele at that marker, the resistant allele, present in that variety, but not in that variety? So we've associated the resistant allele at each of the markers relative to each of the wheat varieties within the panel. So in addition to the panel, he said, okay, we found all these marker trait associations, but you really need to validate that they are real resistances. And we did that within this study by looking at biparental populations. So the breeders very kindly made a range of biparental populations, selecting some of the varieties from the panel, crossing them to susceptible wheats. These populations were screened between 2018 and 22. The populations were genotyped by the breeders, and most of the initial mapping of these biparental populations was also carried out by the breeders. Unfortunately, our GWAS analysis was nicely validated. We have here a number of populations. For example, RAGT made a population reflection times Dickens, and when they screened that for yellow rust, they did a QTL analysis for that. We could validate at least 5 of the marker trait associations that we'd identified in the GWAS panel. So I think in total we managed to validate with these limited number of biparental populations something like 15 of the marker trait associations that we identified in the GWAS. So in addition to the biparental populations, the breeders made a number of near-isogenic lines. Excuse me. So these near-isogenic lines are slightly more complicated. So previous to the Yellowhammer project, there was another project called Wagtail, which was a GWAS study of a number of diseases, including yellow rust. In that study, a resistance marker trait association hit 1, which corresponds to the marker trait association 2A019, which we discovered in Yellowhammer. A hit 2, which corresponds to a marker trait association 6A612 that we also discovered in Yellowhammer. And a hit called 45, which corresponds to 2D99 in Yellowhammer. Okay? So these hits, 1, 2, 4, 5, were discovered in the previous project. So the breeders had already begun to make near-isogenic lines for these hits. Okay, so lines that differed, lines that had the hits and lines that didn't have the hits. Okay, but the background was hopefully pretty much very similar. RAGT also made neoisogenic lines, and in their neoisogenic lines they had lines with and without hit too. Okay, so basically two lines genetically similar except for the hit. One line has the hit, one line doesn't have the hit. RAGT also made neoisogenic lines for a known resistance gene, YR15. Okay, so we had this material, and the reason we wanted this material is because we wanted to look at the phenotypes expressed by these sources of yellow rust resistance at a microscopic level. Rather than just looking at the resistance in the field, we wanted to inoculate these lines and then look at the development of the yellow rust pathogen down the microscope to see if the different sources of resistance affected the development of the yellow rust pathogen differently. Okay? Big experiment, glasshouse experiment. We grew all the lines to flowering. We inoculated the flag leaf of each of these plants with a single isolate collected in 2019. This again shows the value of the survey because we have an isolate that was collected and characterized by the survey that we could use for this research work. We collected the flag leaves, the inoculated flag leaves, over a time course. So we had flag leaves that hadn't been inoculated, flag leaves that were inoculated, that were collected 1 day after inoculation, 3 days after inoculation, 7 and 14 days after inoculation. These flag leaves were then prepared, they were cleared of the chlorophyll, they were stained, and so that we could visualize down the microscope the development of the yellow rust pathogen. We optimize a scoring system. Now, for those who don't know, so yellow rust is a fungal pathogen. The spore lands on the leaf, it produces a germ tube. The germ tube goes in search of a stomata. It enters through the stomata, and underneath, once it's inside the leaf, it forms a substomatal vesicle. From that substomatal vesicle, you get, in the case of yellow rust, 3 infection hyphae, 3 growths that come off the vesicle, which then go in search of a plant cell. When they come in contact with a plant cell, the end of the hyphae differentiates into what's known as a hostile mother cell. It produces a peg that crosses the plant cell wall and pushes back the membrane of the inside the cell, the plasma membrane inside the cell, to produce a fungal hostoria. Now, unfortunately, this staining technique doesn't pick up the hostoria, but the hostoria is the fungal body that grows inside the plant cell, keeping that plant cell alive. It doesn't kill the plant cell because it feeds. It feeds off that plant cell and transfers a nutrient from the plant cell into the hostoria, into the fungus, which then produces these runner hyphae, which then grow between the cells, producing more hostoria mother cells, more hostoria. And this is why in yellow rust you get this striping effect. So from one spore, that one initial infection, you can get these long hyphae that grow up the leaf, producing lots of feeding bodies, and you get this stripe effect. So after 14 days, the fungus then produces a pustule. You get vertical hyphae grow up with spores on them, and that's why you get this stripe of pustules. And that's why Amelia always selects one stripe off a leaf, because she knows that's come from one spore. Okay? So it's a bit complicated, I know, but that's the stages that we looked at. We looked at how many spores that had landed on that flag leaf at each time point, how many had germinated, how many had produced substomatal vesicle, how many had produced infection hyphae, how many had produced runner hyphae, how many had produced pustules. Okay? So, and we did that with the neoisogenic lines that the breeders had developed for us. So the Lima grain lines, using Cas markers, we could confirm that the lines had RRR, had hit 1 and hit 2. Okay? Unfortunately, hit 4.5 was fixed in all the lines. So at the end of the day, we couldn't look at the effect of hit 4.5. We could only look at the effect of hit 1 and hit 2 on this microphenotype. And that's just pictures of it. Okay. So if we compare the line RRS with SSR, so these are two neoastrogenic lines, genetically identical, as much as they can be, but they differ for hit 1. So this line has hit 1, that line doesn't have hit 1. Okay? So this is the resistance that corresponded to the 2A 019 marker trait association found in yellowhammer. And the take-home message is that hit 1 appears to primarily stop the yellow rust development at stage 4. Okay? So stage 4 was the formation of runner hyphae. So this resistance appears to, in some way, slow down or reduce the development of runner hyphae. Okay? If we look at the neoastrogenic lines which differ for hit 2, and I'll explain to you in a minute why I've now called this hit 2.1. So there are lines that have hit 2 and lines that don't have hit 2. We see here that in these lines, the lines with hit 2, but they don't have hit 1, so they only differ for hit 2, can actually let the pathogen can actually develop a bit more. And the hit 2 only stops pathogen development just before pustule formation. Okay, so there are subtle differences in how these two sources of resistance impact on the development of the yellow rust pathogen. Okay, so then we had lines that were made by AHDB, which also, we thought, differed for the same hit. They had hit 2, lines with hit 2 and lines without. These lines were made from a different cross. So in the case of the Limagran lines, the hit 2 came from Belgrade. In the case of the AHDB lines, the hit 2 came from Option. But we discovered using Cas markers that actually the hit 2 in the AHDB lines was different from the hit 2 in the Limagran lines. So hence we called this hit 2.2. So this is a bit of unknown. And we'd already determined based on the phenotype that they did look different. So from the tests of these lines in the field, we suspected that what we thought was hit 2 in these two sets of neoastrogenic lines was not the same gene. However, we continued, we used them in our study, and you can see here that if you look at the microphenotypes, the hit 2 from AHDB, when you look at the microphenotypes, from the early stages of development, doesn't seem to affect the development of the pathogen. And yet we know that this 2 does stop pustules from forming, so it would appear that whatever this gene is doing, it's coming into effect later in the development of the pathogen. And the pathogen can grow and infect and develop, but it's stopped at some point in its ability to form pustules, which is different from hit 2.1, where the pathogen's development was stopped at or retarded at earlier stages. Okay? We then also had 2 lines that were developed by AHDB which differed for the known yellow rust resistance gene, YR15. And you can see that this is a major seedling resistance that has been studied quite extensively. When we looked at the microphenotype between the neoastrogenic lines with and without YR15, again, similar to hit 2.2, we didn't find that the early stages of the pathogen's development were retarded by this resistance gene. But yet we know that it's a very effective resistance and it stops sporulation, stops pustule formation. So YR15, as I said, is a known gene and it's been cloned. So the gene was originally integressed from a wild emmer weed. Okay. It has been used. It was in the GWAS panel. We didn't detect it from the GWAS analysis, because one of the limitations of the GWAS analysis is that any resistance allele that's present at lower than 5% within the panel doesn't get picked up. But we screened the panel with known markers for YR15, because obviously the perfect marker can be developed from the gene itself. So when we screened the panel, we found that the YR15 gene was present in about 2.3% of the panel. So it is used by breeders, it is within the germplasm. But it also gave us an opportunity to test another theory we had. So as part of that big inoculation experiment in the glass house, we collected flag leaves which we used for the microscopy. We also collected flag leaves at each of those time points for RNA extraction and RNA sequencing. So we wanted to see whether, in these lines that differed for a given resistance gene, if we look at expression of wheat genes in that line after infection with yellow rust, what do we find? So I'm only going to show you the results from the YR15 near-isogenic lines. So RNA-seq analysis, we had, okay, it's complex. So we had lines that were uninoculated. So the two lines, with and without YR15, uninoculated, control. We had lines with YR15 1 day, 3 days, 7 days, 14 days after inoculation, and the same for the line without YR15, 1, 3, 7, and 14 days after inoculation. These dots represent the library that was made from the RNA from each of those time points. So you take the RNA, you make a library, and you sequence that library. So you get a whole load of sequences. You can then take those sequences as a lot, that each batch of sequence represents that library, and you can do what's called a principal component analysis. So it compares the libraries. It's basically to check how good the libraries are. And we found that a couple of interesting things. So, okay, some of the libraries were not sitting as they should do. So we expected the control, the uninoculated control, for the lines with and without 15 to sit together, which they did. What we did find is that the lines, sorry, I don't think my mark, no, does it work? Yeah. So the libraries that were inoculated, were sampled 1 day after inoculation, sat a long way away from the libraries that were sampled 7 and 14 days after inoculation. And we think that this is an artifact of the inoculation procedure. So the thing is, when you inoculate plants with yellow rust, you cover them in plastic bags so you've got high humidity, and you put them at 8 degrees for 24 hours in the dark, which is a bit of a strange thing to do, but that's the procedure that's been developed over the years. So we think that physiological procedure, causes physiological changes, which causes the plants at 1 day to sit over here. Yeah? But by the time you get to 7 and 14 days, the libraries are closer together. In this particular experiment, we found that the libraries at 3 days, something had happened to them. They got corrupted, so we had to remove them, which we did, and that's with the rogue libraries removed. So we then did a very preliminary analysis, taking all those libraries, looking at the genes that were differentially expressed between the lines with YR15 and without YR15. And much to our surprise, the gene that actually is the homologue of YR15, because remember, YR15 has been cloned, we know what the gene sequence is, we know what the homologue is in Chinese Spring, came out as one of the top genes within this differential expression analysis. There it is. So that the gene that was most highly expressed was this one. So 1, 2, 3, 4, 5, 6, 7, 8, 9. I can't count. 9. The 9th most highly expressed gene was the YR15 gene. So if we hadn't have known what the YR15 gene was, we could have actually cloned it. So the question is, if this is the case, with the other neoisogenic lines where we've got hits 1 and 2, but we have no idea what the gene is underlying in those hits, those marker trait associations, could we do a similar analysis with those lines and actually have candidates for those resistance genes? And preliminary analysis has pulled out some very good candidates for hit 1, which are on chromosome 2A, in the region where we know the hit is. But that does need, there's about half a dozen candidates, but that needs further verification. And there are technologies that we can use to test those genes fairly rapidly to see if they are candidates for that yellow rust resistance. But that's another story for another day and another project. So just a couple of take-home messages. So an in-depth knowledge of the sources of resistance and the markers that define those resistances, coupled with the annual results from the UKCP Virulence survey, enables breeders to identify those sources of resistance that are still effective within their germplasm. This in turn obviously then enables breeders to produce wheat varieties which are still effective. And as I say, it appears that a lot of the resistances have been accumulated and almost fixed within the gene pool. And there's some suggestion that even those major R gene resistance, once they break down and are no longer effective in the seedlings, could still have a residual level of resistance at the adult plant stage. And then the RNA-seq study may be a means by which we can get a handle on those sources of resistance by developing neoisogenic lines that differ just for the target gene that we want to uncover. So, as you can well imagine, a whole load of people have been involved in this work, and I hope we haven't missed anybody, and thank you to all of them.